Hi, good evening, everyone. Yeah, maybe we'll wait for uh, 3 more minutes. We can start at 6:10. Okay, then we will start today's session. I will start my screen. I will share my screen. My screen is visible to everyone. Okay, okay. So, in today's session, just let me know if someone's waiting in the lobby to get in the meeting because I won't be able to see what is going on, who all are waiting at the like for to join the meeting. So today we will be summarizing whatever was taught in week 2 lecture videos, and then we'll look at few sample questions. Okay. So when we talk about cellular metabolism, this week two's lecture videos comprised of different uh, by uh, glycolytic cycle, Krebs cycle, all these cellular metabolic pathways, and then how to grow different, like what are the different culture media, how to grow bacterial cells, mammalian cells, etc. Those things these lecture videos covered for this week. So we'll just summarize, go through those briefly. So when we talk about cellular metabolism, It can be of two types, anabolism and catabolism. All right. So in anabolism basically refers to the biosynthetic pathways. All right. That is building something up, and catabolism is basically breaking some complex molecule down into simpler molecules. Okay. And which also release energy. Okay. So like catabolic reactions transfer energy from complex molecules to ATP. Heat is released, energy is released, and simple molecules like glucose, we get simpler molecules like glucose, amino acids, glycerol, fatty acids. All right. And anabolic reactions actually require the energy of ATP to produce complex molecules. All right. And then complex molecules like starch, proteins, and lipids. Okay. Now, when we talk about glycolysis, where glucose is used to produce ATP, glucose is broken down to produce ATP. When we talk about glycolytic pathway, there are two phases. Okay, one is preparatory phase, which basically requires energy, or where the steps comprising of the preparatory phase requires ATP. And then there's payoff phase, which are energy releasing steps, which actually release ATP. Okay. So let me zoom. Sorry. Huh. So this is the preparatory phase where ATP is required. So what is happening here? You have a 6 carbon membered glucose molecule. Okay. One ATP is used to incorporate a phosphate molecule to form glucose 6 phosphate. And this particular conversion is catalyzed by hexokinase reaction. Then this glucose 6 phosphate is sidewise the chemical formula and the structure is there. So glucose 6 phosphate is then converted to fructose 6 phosphate, or rather isomerized to fructose 6 phosphate, where the carbon number remains the same, and this conversion is brought about by phosphohexose isomerase. Okay. And then this fructose 6 phosphate incorporates another phosphate molecule to form fructose 1,6 bisphosphate by the action of the enzyme phosphofructokinase. Now, whenever there is any whenever there is any incorporation of phosphorylating enzymes are usually kinases. Okay, so phosphofructokinase 1 is catalyzing this conversion from fructose 6 phosphate to fructose 1,6 bisphosphate. And then you have fructose 1,6 phosphate. This is again a 6 carbon membered molecules, 6 carbon molecule is broken down into two molecules which are each 3 carbon membered, which have 3 carbons. Okay, glyceraldehyde 3 phosphate or and dihydroxyacetone phosphate, or DHAP. This conversion from fructose, this cleavage from this fructose 1,6 bisphosphate to DHAP and glyceraldehyde 3 phosphate is brought about by triose phosphate isomerase. All right. Next comes, so here two molecules of ATP were used, okay, to incorporate the phosphate group. Next we have the payoff phase, which is basically the energy releasing step, okay, of glycolytic pathway. So now you have 3 carbon molecule, okay, I mean two copies of not copies, I mean two 3 carbon molecules, okay. So glyceraldehyde 3 phosphate, now what happens is this undergoes conversion to 1,3 bisphosphoglycerate by the action of this enzyme glyceraldehyde 3 phosphate dehydrogenase. So basically it loses one hydrogen and gives it to reduces the NAD plus molecule, okay, forming NADH. One NADH for one glyceraldehyde 3 phosphate. For two molecules of glyceraldehyde 3 phosphate, we have two NADH molecules. Fine. Now this 1,3 bisphosphoglycerate, which is again like individually these represent the number of molecules here, and so each of this 1,3 bisphosphoglycerate is also a 3 carbon compound. All right. Now 1,3 bisphosphoglycerate is converted to 3 phosphoglycerate, and it releases one phosphate group to form and joins with ADP, which in further joins with ADP to form ATP. Okay, releasing one ATP molecule. And then also the enzyme required here is phosphoglycerate kinase. And then 3-phosphoglycerate to 2-phosphoglycerate conversion is phosphoglycerate mutase, catalyzed by phosphoglycerate mutase. And then phosphoglycerate, 2-phosphoglycerate is converted to PEP or phosphoenol pyruvate, uh, catalyzed by this enzyme enolase. And then PEP is converted to pyruvate finally to like by the action of the enzyme pyruvate kinase. Okay, so in this um, whole conversion we have one NADH molecule released, one ATP molecule released, and one H2O molecule released, and again from PEP to pyruvate conversion we have one ATP molecule released for one like from one 3 carbon moiety glyceraldehyde 3 phosphate. Fine. Now if there are 2 carbon moieties, everything gets doubled in the end. Correct? Sorry. So when we look at the balance sheet for glycolysis, so input is 1 glucose, uh, 4 ADP and 4 inorganic phosphates, right? And 2 ATP, 2 NAD plus in total, including both the preparatory phase and the payoff phase. And then output is what we have finally is 2 pyruvate molecules, 3 carbon moieties, um, 2 net production of ATP, okay, because 2 are used up and then 2 are like 2 are released here, and 2 net ATP molecules we are getting, and then NAD plus molecules, 2 NAD plus are incorporated, and 2 NADH molecules we get out of this glycolytic pathway. Now the output molecule, the pyruvate molecules are used up for the next metabolic pathway, which is the Krebs cycle or TCA cycle. Now, when we talk about the regulation of glycolytic pathway, what happens is one form of regulation is via the insulin. Okay, insulin and insulin receptors and glucose transporters. So first, when insulin is bound to the insulin receptor here, what happens is insulin binds to its receptor in the cell membrane. Okay, and then this activates this receptor and triggers the recruitment of these glucose transporter molecules in the cell membrane. The glucose transporters are already, which are present already in the cytosol. Okay, these glucose transporter molecules are transported from this intracellular pool to the cell membrane. These are the glucose transporters here, and then glucose transporters increase insulin mediated uptake of glucose into the cells. Okay, these are incorporated here, the glucose transporters, and then glucose is taken up inside the cells. And when insulin levels decrease, glucose transporter moves out of the cell membrane, okay, and comes back to the cytosol, um, uh, toward like joining the intracellular storage pool where they can be recycled further. Okay, this is a type of like insulin mediated regulation of glycolysis. Another regulation of glycolytic pathway is via the phosphorylation of pyruvate kinase enzyme. Okay. Now, when pyruvate kinase is actually what we saw earlier in the pathway also, it actually takes a phosphate from phosphoenol pyruvate to give it to pyruvate molecule. Fine. Now, pyruvate kinase, this particular enzyme has two forms. One is dephosphorylated form, which is more active, and another is the phosphorylated form, which is less active. Okay. So, when there's low glucose in the body or in the cells, uh, what happens is it dephosphorylates the pyruvate kinase from the phosphorylated one, which basically activates the pyruvate kinase enzyme. So, activated pyruvate kinase further catalyzes the conversion of phosphoenol pyruvate to pyruvate, thereby producing energy. Okay, but when there's high blood glucose, so we already have sufficient energy. So glucose molecules, what do? They catalyze, they phosphorylate the like helps in the phosphorylation of pyruvate kinase enzyme. And if phosphorylated pyruvate kinase enzyme is phosphorylated, it actually slows down the conversion of PEP to pyruvate. Okay, this is another point of regulation of glycolytic pathway. Another is the presence of these molecules or substrates which can hinder or boost the conversion at each step. Okay, from glucose to glucose 6 phosphate, um, and then glucose 6 phosphate to fructose 6 phosphate. All these conversions by the presence of wherever these there are these plus molecules, this actually helps in the conversion of that particular, helps in catalyzing the reaction in the forward direction, and the minus ones represent these entities which actually uh, doesn't really promote the forward reaction. Okay, rather inhibitors of the forward reaction. Next we come to the Krebs cycle or TCA cycle. So you have the pyruvate molecule from the glycolytic pathway. Now pyruvate is oxidized and the citric acid cycle or Krebs cycle or TCA cycle takes place in the mitochondrial matrix. Fine. So this is a 3-carbon entity, pyruvate molecule. Then this is oxidized to acetate, and um, coenzyme A is added to the acetate, forming acetyl coenzyme A. Okay. Now what happens is this acetyl coenzyme A again um joins with oxaloacetate. So the 2-carbon acetyl group and 4-carbon oxaloacetate, 1, 2, 3, 4, combine to form this 6-carbon citric acid molecule. Okay. And then the 6-carbon citric acid molecule or citrate is converted to isocitrate and isomerizes to isocitrate. And then isocitrate, this is also a 6-carbon molecule. This gets converted to alpha ketoglutarate, which is a 5-carbon molecule, and this conversion releases one molecule of CO2 and NADH. Okay, and then this 5-carbon alpha ketoglutarate forms um, succinyl coenzyme A, which is again a 4-carbon moiety, and this conversion also yields one CO2, one molecule of CO2 and one molecule of NADH. Okay. So this step is almost identical to pyruvate oxidation. And then what happens here is succinyl coenzyme A is converted to succinate, okay, where it releases the coenzyme A and converts GTP to GDP, which is coupled to the conversion of ADP to ATP molecule. Okay. And then this succinate is converted to, rather oxidized to fumarate, which releases hydrogen, and this hydrogen is taken up by FAD to form FADH2, and fumarate then takes up water to form malic acid, and then this malic acid is oxidized to oxaloacetate, 4-carbon moiety, uh, which can again join with or react with the acetyl coenzyme A, formed one of the early intermediates of the TCA cycle, to form citric acid cycle, citric acid, and then the cycle goes on. So when we talk about TCA cycle, it is occurring in the mitochondrial matrix. There are 2 cycles per glucose molecule, because for one pyruvate molecule, one acetyl coenzyme A molecule, there are 2 molecules of pyruvate that we get from the glycolytic cycle. So for one glucose molecule, there are 2 cycles of Krebs or citric acid and TCA cycle. We get in total 3 NADH molecule from one acetyl coenzyme A, one FADH2 molecule from one acetyl coenzyme A, 2 carbon dioxide molecules are released. Okay, and 2 acetyl coenzyme A transforms into CO2. So the inputs are basically 2 acetyl coenzyme A. For one, this whole chart is for one glucose molecule, 2 acetyl coenzyme A, 6 NAD plus, 2 FAD, and 2 ADP and 2 inorganic phosphates are giving us 4 molecules of CO2, 6 NADH, two FADH2 and two ATP molecules. Okay, ATP is there where it is coupled to GTP synthesis. Okay, now when we talk, so this is uh, the TCA cycle. Now, when we talk about anaerobic respiration is basically either lactic acid fermentation, where you have two pyruvate molecules formed from the glycolytic pathway. You have net two ATP molecules, but then these pyruvate molecules are used up to form lactic acid, thereby using up the NADH that was released from the glycolytic cycle. Okay. Now, aerobic respiration versus anaerobic respiration. So, in the cytoplasm, you have the glycolytic cycle taking place, and then in the mitochondria, you have the TCA cycle. Like pyruvate gets oxidized to form acetyl coenzyme A, then it enters the Krebs cycle, and then you have oxidative phosphorylation. Okay, and then but in anaerobic respiration, glycolytic cycle is happening in the cytoplasm. You have ATP molecules released, but then the pyruvate can either undergo alcoholic fermentation to form ethyl alcohol and release carbon dioxide, or it can produce lactic acid, which is also termed as lactic acid fermentation. Right? Now, when we talk about this, was about the metabolic reactions happening in the body, in the cell. Now, when we talk about growing microbes in any, how do we harvest microbial cells, whether be it mammalian cells, yeast cells, or bacterial cells? You have to provide them the nutrient proper appropriate nutrient conditions. Okay, so that is what is called as culture media. So culture medium is the mixture of organic and inorganic materials in which microbes are grown. Now, culture media can have different categories and classifications. Mostly, like majorly, these are classified based on what is the physical state of the medium. It can be liquid or solid. What is the composition? What are the constituents of the culture media? So there are different. I think you all must have gone through the lecture videos also. So they based on what is the requirement. To like how are we going to use the microbes for further processes, further downstream processes? Based on that, the composition can also vary, and also what is the function of the culture media. Based on that, the culture media can be classified. Okay, so on the basis of physical state, it can be liquid, which is also this should be broth. Okay, broth. Liquid medium is also called as broth, which is without agar, and then semi-solid medium. These have small quantity of agar, and which gives the whole culture medium a gel-like consistency. Okay. Usually, whenever researchers want to check the motility of microbes, like how, um, suppose they have modified the microbe, or they want to see how, now they want to really check its motility, that how, suppose a certain change in their some kind of molecule or some kind of gene involved in the neuronal pathway, suppose, is affecting its motility. So what people usually do is they use semi-solid medium, where the microbe can easily move through. The liquid medium is like they can monitor the movement of the microbe through that particular medium. Liquid medium is like you can't really monitor it properly as such, and solid medium, it would be too thick and viscous, and the matrix would be too cross-linked for the microbes to really get in through and move. The motility will be hindered. And then you have the complete solid medium, where agar is added. It can be 3% or 2% agar as well. Okay. So it is used to check the morphology of bacterial colonies. Fine. And then on the basis of the composition of the culture medium, you have chemically defined medium, where we really know the exact chemical composition or chemical constituents of the particular culture medium in question, and it has chemically pure organic and inorganic specific compounds according to the nutritional requirements of microbes. It is glucose salt broth. It will be broth where we know the exact amount of glucose. This is an example where we know the exact amount of glucose, exact amount of salt in it. Okay. Then there's complex media also, where there are like different animal extracts, plant tissue extracts, or basically it's a little complicated, which are required for certain growth conditions of certain microbes. For example, there's yeast extract agar. Now, on the basis of the function, also we can divide the culture media into general purpose media, which supports the growth of a large number of microbes. For example, in general, any kind of nutrient agar. And there's enrichment media, which contains substances which actually favors the growth of certain specific microbes only. Okay, for example, blood agar. Now, this is from the lecture video only, lecture slides. So, what are some of the common media constituents for bacterial growth? There are amino nitrogen constituents where you have peptone, protein hydrolysate. There are growth factors in the medium, which can be taken from blood or serum or yeast extract or vitamins. There are it needs to have energy source definitely, which comes from sugar, alcohols, or carbohydrates. And there are salts or uh, buffering agents, which comes from phosphates, acetates, and citrates. Then there it needs to have minerals and metals also. It also has to have some selective agents. Okay. Why selective agents? Because suppose we want a particular type of bacteria. Okay, we have modified the bacteria to produce a gene of our interest. Now, other bacteria are also there in the surrounding. So, if we are culturing in general without any selection, that okay, so there are certain selective agents added in the culture medium so that only those bacteria that are producing a gene of our interest or producing a protein of our interest is only selected out of the different kinds of bacteria that are already prevalent in the surrounding. Okay, so if you give in general a culture medium, a rich culture medium, any kind of microbe will find it, very it will be able to thrive on that. Okay, it will have good amount of resources, and even the bacteria of our interest can be outcompeted by some other bacteria. We don't want that. So that's why we usually have selective uh, like selective stress, so that only our uh, I'm just giving an example of bacteria here, so that the bacteria containing our gene of interest is amplified, or is able to divide. Others are not able to, or others are killed. Okay, that happens. We will eventually look also in the subsequent lecture videos, that how are these happening, what is like people do put in antibiotics. There are all these selective stress, or selective colors to select out the bacteria of our interest. Okay, then there are indicator dyes also, phenol red, neutral red. And there are gelling agents. We talked about agar. There are also silica gel, gelatin, alginate. Okay, now these are some examples of bacterial growth medium, which can be either like enriched media or nutrient limiting media. So M9 minimal media, these are like limiting growth medium. Okay, and LB media, SOB, terrific specially is like highly enriched nutrient medium for bacterial growth. Okay, so these are few examples and the compositions and the applications written over here. Mostly, sorry. Haha. Okay. Mhm. Haha. So it is basically the types of medium. These are all company made media for different bacterial growth. Okay. So what happens is different bacterial growth medium. So these are named based on the composition. It has different kinds of salt composition, phosphate composition. There are extracts added to it. Okay. And it has different kinds of applications, mostly for bacterial growth only. Okay, bacterial growth, bacterial plasmid production, and bacterial protein, like bacteria are also used for protein production. Okay, so there are different kinds of medium where it has a large amount of nutrients in it, or it can have minimal amount of nutrients in it. Okay, so based on that the composition varies. This is what the basically the table is showing, just summarizing what all medium, culture mediums are available for bacterial growth. Okay. Now, the previous table was about bacterial culture growth medium, like bacterial growth culture medium, and these are for yeast medium, okay, for culturing yeast cells. So again, these are the names of the media, okay, and these are the composition, what is actually found inside those culture mediums, and what are the applications. When do we use this particular type of medium? Okay, so this is also in your lecture slides also for the lecture videos. And what happens usually is, whether while preparing the yeast culture medium or the bacterial culture medium that we saw earlier, what we usually do is, it has because it has these compositions, certain amount of salt, certain amount of glucose, certain amount of phosphates, etc. So what happens is, they have a predefined, suppose this many grams of powder will go into distilled water, and that is how you basically prepare the media. Okay, liquid media, I'm talking about the liquid media as well as the solid media also. And then you have to autoclave the stuff. We will come to autoclaving and sterilizing, so that we have to sterilize these culture media so that we don't contaminate it with any other microbes. Then there's mammalian cell culture medium. What happens here is, it has um, it has this particular um, salt, there's serum in it, there's antibiotic in it, so that you don't uh, contaminate the mammalian cells from other bacterial or yeast cells. Okay, so what we have to take care of it, because mammalian cells require maintain a certain range of pH. Okay, pH of the media has to be taken care of. Then it has to be filtered to get rid of the contaminants, and filtration should be performed at low speed. So why is it so? Because filtering is basically, you have it's basically a filter only having different kinds. The pore size is as small as 0.22 micrometers. Okay, so through those filter, there are filter papers having pore size of 0.22 micrometers. So you are basically filtering your whole culture media through those filters, and it has to be at low speed, because what happens is, when you are actually filtering, uh, you don't want to clog the pores. First of all, you also don't want to, when you are giving too much pressure, it can happen that some contaminants are forced through the pores. Okay, so we don't really want that. So filtration is always performed at low speed. Then you have heat inactivated serums. Okay, serum is having different kinds of proteins or antibodies rather. Okay, so which has to be heat inactivated. Okay, so that it doesn't really uh, interfere with the culture, mammalian cells, like propagation of mammalian cells. Then there are antibiotics to help prevent any contamination from bacterial or fungal cells, like any kind of bacterial or fungal contamination. Okay, and so there's also usually some kind of pH indicator, phenol red, added to these culture media, which changes color when the pH of the medium changes. Okay, then you know that, okay, it there is some change, like it has become too acidic or too basic, something of that kind, so that this you immediately discard it and then start afresh. Okay. Now, what are the common types of cell culture media? These are, again, these are company names. Just whatever company has started preparing these media, this is what it's named after. So just to give you a flavor of how it looks like, what are the names predominant in the market, that's why I've made this table. This is just for a brief exposure of what are the names and all. So EM, Eagle's Minimal Essential Medium, was first developed in 1959. Okay, And then slowly all these modifications came into the picture, where concentrations, like higher concentrations of vitamins and amino acids were taken. And then there's this particular medium where it's more complex, which is usually used to culture human lymphocyte cells. Okay. And then there's IMDM, which is again modified version of DMEM for high density cultures. And there's McCoy's 5A, Ham's F12. Okay. These are all from, like, basically for cancerous cell lines. Okay. So these are just examples, and these are all company names. Okay. Which basically prepare these media and supply in large quantities. Now, what are the precautions that have to be taken while preparing bacterial growth media? I was talking about sterilization techniques. Fine. So what happens is, you are first preparing you have the composition given, you are preparing your culture medium or growth medium, and then you have to sterilize it. How do you really sterilize it? You sterilize it using the using something called autoclave. Okay. Autoclave machines are either wet sterilization or dry sterilization. In wet sterilization is autoclaving, where you basically put these media. Okay, first you are preparing it in some kind of culture flasks or conical flasks that you might have seen around. Okay, so those culture flasks are first cleaned properly, then you put in your water and the growth medium powder, whatever comes along with the preparation, according to the specified preparation protocol. And then you put it inside autoclave, where basically the pressure, you really autoclave it using the steam coming out from distilled or double distilled water. Okay. And that pressure, like 121 degree Celsius heat and 15 Pascal pressure, what happens is you are wet sterilizing the um, culture medium. And then what happens is there are other stuffs to be used during culture medium. There are pipette tips, micro tips, or suppose some tubes or any kind of other culture medium tubes, or even these glass flasks or glasswares are all kept back into ovens, basically, which reach up to 70 to 80 degrees Celsius for dry sterilization. Okay. And so these are two types of sterilization techniques that are taken care of while preparing, that has to be done while preparing any kind of growth medium. Okay. Specifically bacterial growth mediums. And also, one thing to be noted is, suppose um, you have to wash the flasks. Now, while washing the flasks, usually people use detergents and all. Now, there shouldn't be any remnant of detergent on the flasks, so that it actually uh, like prevents bacterial growth. It actually lyses the cells. Okay, so we have to be very particular about washing the flasks, uh, baking it inside hot air ovens. Hot air ovens are basically those which have 70 to 80 degrees Celsius temperatures for dry sterilization. Okay, so autoclaving, or in general, autoclaving is wet sterilization, and hot air ovens are for dry sterilization. That has to be done during preparation of growth medium. Okay. Now, when we talk about the division of bacterial cells, they predominantly undergo binary division. What happens here is, the cell replicates its DNA, the cell wall is the same, cytoplasmic membrane is the same, and this is the replicated DNA here. And then the cytoplasmic membrane elongates, separating the DNA molecules. Then there's a cross-wall formation here where the membrane is actually invaginating, and then the cross-wall forms completely there, and then it divides into two daughter cells. This is the predominant way of division in bacterial cells. Another is budding form. In bacteria and yeast also, there's budding type of division, where the bud begins to form on this is the parent cell with the nucleus. Bud begins to form, and then nucleus copies and divides, the bud receives one copy, and bud becomes a separate cell altogether, and then chains of daughter cells are produced. Okay, and then there's fragmentation. Fragmentation mostly happens during any kind of unfavorable conditions. Okay, this is the protoplasm of bacteria which undergoes compartmentalizations. Okay, it is divided into these compartments here, and then these are fragmented into these small bodies here, each fragment called gonidia or gonidium, one fragment gonidium. And when the favorable conditions return, each fragment or each gonidium grows into a new bacterium. Okay. And then definitely prior to fragmentation, the bacterial genome has to undergo repeated replication so that each fragment can get a copy of it. Okay. Now, when we talk about a bacterial growth curve actually follows a distinct like shows a distinct growth curve where each phase of this growth curve is divided based on how is the whether when is the bacterial cells preparing for its division, when it is actually dividing actively, and when it has when it has started dying, and when it's finally when the rate of death and the rate of division are equal, or when one is compensating the other, or one is sup- like uh, bypassing the other. Okay, so the first phase, when we plot a bacterial growth curve, it has the number of logarithmic scale number of bacterial cells, and which is the dependent on the time period. Okay, so first is the lag phase, which is the very initial phase here, this part, and which is characterized by cellular activity, but there's no growth in there or no division is there. Cells are increasing in size, definitely because they are preparing themselves for uh, final division. Okay, they have to replicate their DNA and prepare all those enzymes, factors, etc. Okay, but there's no actual cell division taking place, and cells are busy synthesizing a large amount of protoplasm before moving on to the next phase of active multiplication. Okay, so this is the lag phase, and then you have this part, the exponential phase. Okay, or exponential phase is also known as log phase, where cells are actively dividing and doubling in numbers after each generation time. Okay, so the typical generation time of E. coli bacteria, which is most commonly used for laboratory experiments and experimental studies, uh, very common model organism, is only 20 minutes. Okay, so during log phase, cells are actively dividing and doubling, and when their metabolic activity is really high, what is happening here is DNA, RNA, cell wall components, and other substances which are required for growth are generated for division. And during this time, during the log phase, antibiotics and disinfectants are most effective. So bacterial cell walls or transcription and translation processes are targeted. So what basically means is during now, uh, suppose one how do we when do we really want a bacterial, because bacteria are uh, both pathogenic as well as non-pathogenic. Usually people in the labs work with non-pathogenic strains, they also work with pathogenic strains. So against pathogenic bacteria, when you really want to target bacterial cells, usually this particular phase of bacterial growth is targeted. The antibiotics or disinfectants are most effective during this particular phase when the cells are actually dividing. So bacterial antibiotics or disinfectants target the cell walls of bacteria, or the protein producing machinery of bacteria, where they are actively dividing. So if you're targeting those particular crucial processes or crucial machinery, or the cell wall itself where it is not able to make the cell wall only, so what happens is the division is inhibited, and if the division is inhibited, ultimately the growth is inhibited. Okay, so this is the phase where mostly these antibiotics and disinfectants target the cells. Okay, next comes there are resources available already during exponential phase, so they are bacterial cells are very happy, they are using the resources and dividing, having good amount of food, and dividing themselves. But during stationary phase, what happens is it actually reaches um, a asymptote, or rather a plateau phase. It reaches a plateau phase where the growth declines because slowly they have rapidly divided in the earlier log phase. Okay, they have used up a lot of available nutrients and resources. Now the competition for nutrients will slowly increase, right? And then also their division will lead to, as more number, like the number of cells increases, what happens is waste products will also start to accumulate. Then the cell growth will actually reach a stationary phase where the number of dividing cells will equal the number of dying cells. So overall population growth we won't be able to see. Okay, and cells slowly become less metabolically active if we talk about the whole culture there. And then spore forming bacteria also produce endospores because spores are also usually produced during unfavorable conditions. So as the nutrients start getting limited, okay, as it gets, or waste products are accumulating slowly, then bacterial cells resort to spore formation. And also pathogenic bacteria then start generating substances which are also called virulence factors to survive harsh conditions, and then can cause diseases as well. Okay, next comes death phase. Definitely what happens is the number of or death rate or number of dying cells increases, nutrients are less available, and accumulated waste is much higher, and number of living cells decreases exponentially during this time, and there's a sharp decline in growth. Okay, so this is basically lag phase, when the cells are preparing itself for division. Log phase or exponential phase, when bacterial cells are actively dividing. Stationary phase, when the bacterial cells have started dying as well, the nutrient conditions are getting limited. So number of cells dying is equal to the number of cells dividing. And then the death phase is number of dying cells increases because the nutrient has become really limited, and waste production, like waste accumulation, is quite high. Now, how do we measure the microbial growth? While measuring microbial growth, there are different ways to do that. One is, first is microscopic count. You basically have the suspension of cells. You put it under the microscope and basically manually count it out. Okay, so how many cells are there in the particular suspension? Okay, so one is use pour plate or spread plate method for viewing under a microscope. You directly pour one, suppose 1 ml of cells on a petri dish, or 500 microliter of cells on a petri dish. Simply put it under the microscope and count the number of cells visible. Okay, then there's definitely one thing that should be taken care of, that concentration shouldn't be too high, because what you will eventually have is too dense, or a lawn of cells there, overlapping cells. You won't be able to count, like distinctly be able to say that, okay, this is one cell, this is the other cell lying beside. So, or if it's too few in number, it will be insufficient to really count and get the whole number of cells in the complete original culture. Okay. So this is what I was referring to. You are physically counting the number of bacterial cells under a microscope. Okay. So what usually is done is serial dilution and plating. So you have the original culture. It has, like, it is really turbid because the cells have grown to really exponential numbers there in millions of copies. So this is really turbid. Now what happens is, uh, suppose from the original inoculum, you have your culture medium, 9 ml of culture medium, and 1 ml of your original inoculum. Okay, and then you plate it out. This is the liquid medium, liquid culture medium, and this is the solid culture medium, which is the agar. If you plate out, this is 1:10 dilution of the original source, and then when you plate it out on the culture medium, you have a lawn of cells, like a complete smear, so you don't have individual bacterial colonies to count. Okay, then you again dilute it, again 9 ml of liquid medium, culture medium, and 1 ml of the 1:10 dilution. So you basically have 1 is to 100 dilution. You again plate it out on a solid medium. So you see that the whole smear is slowly clearing up, but still you don't really have distinct colonies, individual colonies to count. Then again, you basically make the dilutions until you really have, like suppose you can distinctly make it out here that there are 4 colonies. Here also you can count it, but now when you are finally measuring that how many cells are there in the original source or original inoculum, you have to take into consideration this dilution factor. Okay, so you have diluted it by 10 to the power 5 times here, right? Sorry, 4 times it. 10 to the power 4 times here in this example. So then if there are 4 bacterial colonies here, and on this particular plate you are getting 4 bacterial colonies after diluting it by 10 to the power 4 times, then what you have is here you will have 4 into that many number, like 10 to the power 4 times of cells in the original inoculum. Okay, here in this example, suppose here they have like 32 colonies here, it is 10,000 here, so 32 into 10,000. If it would have been 4, 4 into 10,000, dilution factor has to be taken care of. And also one more thing, how much are you putting in for plating? So here, this example, they have used 1 ml of culture to plate it. If you are using 500 microliter of culture to plate, after each dilution, you have to take into account that much amount of culture volume into consideration while calculating the bacterial cells in 1 ml of sample, 1 ml of culture medium. Okay. Another is you are measuring the turbidity of the sample. Okay. So what happens is this is your the vial, culture vial, and you have the media there, and you let light pass through the culture tube, and you measure the the light rays that pass through the medium, through the culture medium, is detected on a detector, and then the readings are taken. But if there are some cells inside, so what will happen is those cells will actually hinder the light passage, okay, passage of light through the medium. So what will happen is the amount of light passing through this particular medium, if there are bacterial cells suspended all over the culture medium, you will have that it will detect less amount of light. Okay, and so it will give a corresponding reading there. Now what happens is this complete measurement is done using an instrument called spectrophotometer. Okay, there is a light source here. Now when you are taking readings for a culture medium, fine, for the growth of bacterial cells, what you have to do is you can't really keep on taking only the values corresponding to different bacterial suspension. You have to have a reference, right, from where you will be comparing that, okay, this is how much. You suppose you have a 0, then you know that, okay, this is 1 greater than this is 2 times greater than this, right? You have to have a reference point from where you will be comparing the growth. So what ideally people do is whatever bacterial cells are suspended in the culture medium, okay, that culture medium only without any bacterial cells are first, the reading is first taken, and this particular reading is called a blank measurement. Okay, blank means there's nothing in it, no cells in it, only the culture medium. Now if the suppose if, not only the culture medium, if the cells are suppose resuspended in any kind of buffer, so that only buffer without the cells are first, that buffer reading is first taken, and that is the blank measurement that is taken as the reference reading. And then you start measuring your uh, cell suspension, whether it is in buffer or in culture medium. Accordingly you take the blank measurement and then start measuring the turbidity of your sample. Okay, and then one can also start measuring the wet and dry mass. Wet mass is basically you have water content in it, and dry mass is you completely remove the water from there. You have the only, like, without water, you have only the dry mass or of the cells that you have harvested. Okay, so one more thing. This is all bacterial suspension we are taking in. Now, when we talk about wet and dry mass, so how are we having the cells? So, in the previous tutorial class, we saw we actually centrifuge, right? We centrifuge to get the cells, the hard or the heavier substances pellet down, and you get a supernatant. Fine. So the cell pellet, this is how the cells are harvested. You have to have to centrifuge it at around little low speeds as compared to separating the components of a cell. So if you want the whole cell to be pelleting down, so you can do it at between 5000 to 6000 rpm for 10 minutes for suppose a 1 liter culture or 30 ml culture, and for 2 ml cultures or 1 ml culture, 2 minutes centrifugation at 6000 rpm is more than enough to harvest the complete cells. We are not breaking down the cells here. Okay, we are just separating the complete cells from the culture medium, and we are harvesting the cells down, and then we can process it for any kind of downstream processes. We want to purify plasmids from those cells, if you want to purify DNA from the cells or RNA from the cells, whatever is the downstream application, if we want the cells first, we have to centrifuge it. Okay. So this is how once we harvest the cells, we can then calculate the weight of the cell pellet that we have harvested. Okay. So now moving on to the questions here. Which hexokinase we saw it in the uh, glycolytic pathway also. Which of the following is the cofactor of hexokinase? The answer is magnesium here. So just to give you an idea that there are enzymes which catalyze chemical reactions. Now some enzymes require cofactors to be to be really active and perform its activity, to be functional. Okay. So if the enzymes are basically proteins. Now if the enzyme is protein only, then it is it doesn't really require the cofactor. So enzyme is protein only, which is lysozyme. Okay. Now cofactors are non-protein moieties or non-protein entities of an enzyme which helps it to become functional. Now cofactors can be organic molecules. In that case, it is coenzymes, or it can have inorganic ions. For example, in case of hexokinase, magnesium, like divalent magnesium ions are the cofactors here for hexokinase. Okay. Uh, Another example is cofactors can also be permanently attached. In this scenario, it is called a prosthetic group, okay, without which the enzyme won't be functional. And there can be temporarily attached moieties onto the enzymes to make it functional during a particular reaction, during when the enzyme needs to catalyze a particular reaction, and then it can detach itself from the enzyme to participate in or to help in some other reaction or some other enzyme. Okay. So this is what is called cofactor. So for this particular question, cofactor of hexokinase is magnesium 2 plus. Okay. Now in glycolytic pathway, we saw two molecules of ATP being utilized by the enzymes dash and dash. So if anybody remembers or recalls, can someone answer? So the answer will be, this is the preparatory phase. So ATP molecules are getting utilized here. Okay. So the enzymes that are utilizing it to convert, one is glucose to glucose 6 phosphate. Phosphate is getting incorporated, like phosphate is incorporated into the molecule. So we have kinase enzymes here. Whenever there are like ATP is getting utilized, you have the kinase enzymes here. So one is glucose to glucose 6 phosphate, hexokinase, and then fructose 6 phosphate to fructose 1,6 bisphosphate. You have phosphofructokinase. So answer is hexokinase and phosphofructokinase enzymes. Now in the previous picture only we saw fructose 1,6 bisphosphate, which is a 6 carbon molecule getting broken down into glyceraldehyde 3 phosphate 3 carbon molecule and DHAP, which is also a 3-carbon molecule. So the enzyme used is, does anyone remember? The answer is aldolase enzyme. Okay. Now, which of the following cell surface receptor is involved in the uptake of glucose? Uh, so the answer is option C, glutes. Okay. Now, when there's low blood glucose, what happens? So we looked at regulation of glycolysis by the enzyme, by the phosphorylation or dephosphorylation status of the pyruvate kinase enzyme. We saw that when the blood glucose levels are lower, then the enzyme pyruvate kinase is phosphorylated. If we look at again the pathway, just to brush up, what happens is pyruvate kinase enzyme helps in the glycolytic pathway for the conversion of PEP to pyruvate, and it has two forms. One is the less active phosphorylated form, and the another is non-phosphorylated or native form, which is more active. Now, when under low glucose conditions, pyruvate kinase is dephosphorylated, which helps in the conversion of PEP to pyruvate and produces energy. But high glucose conditions, what happens? Um, the pyruvate kinase is phosphorylated and it slows down the conversion of PEP to pyruvate. All right. Now, one molecule of GTP is equivalent to dash molecule of ATP. One molecule of GTP is equivalent to one molecule of ATP. Option is A. Which of the following pairs positively modulate the conversion of isocitrate to alpha ketoglutarate? The answer is ADP and calcium. Okay. If we see the conversion of isocitrate to alpha ketoglutarate is actually carried out by this enzyme isocitrate dehydrogenase. It is like reducing the NAD plus to form NADH, and this particular enzyme isocitrate dehydrogenase is activated by ADP, citrate and calcium ions, and it is inhibited by the presence of NADH, NADPH and ATP. Okay. Now, production of alcohol under anaerobic conditions in yeast is facilitated by the enzyme alcohol dehydrogenase, which converts acetaldehyde to ethanol. The substrate pyruvate binds to the active site of this enzyme with the help of a dash ion. So basically, what is, if we straightforwardly could ask the question, it is what is the cofactor for alcohol dehydrogenase enzyme? The answer will be option C, zinc ions, Zn2 plus. Okay. Next is, which of the following is the function of phenol red in DMEM? DMEM is the mammalian cell culture medium. Now we looked at the pictures previously that mammalian cell culture medium is pink in color. Okay. So in general, we talked about some kind of pH indicator, which is phenol red in this case, which helps to detect if the pH of the medium is higher, like more acidic or more basic. Then one can play around, or one can even start afresh, add fresh culture medium to help the mammalian cells thrive. Okay. Um, so phenol red is basically to detect the changes in pH. Okay. Now, this is when we talk about counting the number of bacterial cells in a culture, this is another Petrov. Huh? What? Huh? This question you want me to repeat? Okay, sure. Okay. Now you have something called a counting chamber, Petrov-Hausser counting chamber, where you count the bacterial cells. Okay. So what happens is you have a suspension of cells. All right. Now these are plated here, not really plated. These are spread over here on this chamber. Now these chamber have characteristic dimensions. What happens is the chamber has a square of equal volumes. Now this is basically a glass slide, these ones. Okay. Now the chamber is having uh, an area of the squares are having an area of 1 by 400, and the height of the squares is 0.02 millimeters. So you have this particular volume. So each chamber is having 1 by 20,000 millimeter cube of volume. Okay. Now when you have a bacterial suspension, you put in a bacterial suspension, cover it with a slide or cover slip, and then you count the bacterial cells under a microscope. Now, what happens is, if suppose you are having, you are able to count 8 bacterial cells in this particular chamber, then what happens is, now the chamber's dimensions are calculated in millimeter cube. Okay. Now, 1 millimeter cube, okay, uh, 1 millimeter cube is there, and each square or the volume that it can contain is 1 by 20,000 millimeter cube. So if you are able to count 8 bacterial cells in this particular square chamber, then you are having 8 into 20,000 into 10 to the power 3 number of bacterial cells per ml. This 10 to the power 3 comes in picture because millimeter cube has been converted to ml. Okay. And then again, what we talked earlier, that if the cells concentration or the culture medium is too concentrated, you can't really calculate the number of cells. There will be overlapping cells. You won't be able to make it out. Like, you won't be able to distinguish different cells from each other. And if it's too low, then suppose you have like only one cell, that is not a very good count in this scenario. So you should ideally be having some 8 cells, 10 cells, 30 cells, like that, so that you are able to count distinctly how many cells are there, and you have an idea of the dilution factor also, so that you take it into account as well. So the next two questions are about this chamber only. So in a Petrov-Hausser counting chamber, the volume of each chamber is, this is a straightforward answer. What we looked at is option C, 1 by 20,000 millimeter cube. Now, based on the previous example, if each chamber which is having 1 by 20,000 millimeter cube of volume, now you are able to count 5 bacterial cells in each chamber. Okay. Then what is the total number of cells per ml of the culture? Okay. So if it is 1 by 20,000 per 1 by 20,000 millimeter cube of volume, you have 5 bacterial cells. So it will be 5 into this much volume into the into 10 to the power 3, right? So you will have 5 into 20,000 into 10 to the power 3. So what will be the answer is 10 to the power 8 number of bacterial cells per ml of culture. This is just an example of how to solve this particular question. Okay. Now, this is a straightforward question where uh, it's asking the correct sequence of different phases of bacterial growth. So what we saw is, first it goes through a lag phase, then it reaches an exponential or log phase, then it reaches a stationary plateau phase, and then the cell growth declines, which is the death phase. Okay. So option is B. Option B, lag, log, stationary, and then death phase. Now, in which of the following phase newly divided bacterial cells derive nutrition primarily from the older dead bacteria in the culture? So, in lag phase, there are no dead bacterium as such because all the cells haven't divided yet and they are preparing for division. Okay. In log phase, cells are actively dividing. In stationary phase, the number of dividing cells and the number of dying cells are equal. Okay. So nutrients start getting limited. So what happens is, the newly divided bacterial cells can replenish their nutrient requirement from older dead bacterial cells, but up to a certain point, definitely, because after that you will have like a death phase where even the nutrients from the dead bacterial cells are not enough for the newly divided cells. Okay. But to answer this particular question, newly divided bacterial cells derive nutrition primarily from older dead bacteria in the culture during the stationary phase. Option C. Okay. Now, this is a question here which talks about a bacterial culture medium was inoculated with roughly 5,000 E. coli cells at 10 a.m. in the morning, and it was allowed to grow for 12 hours. Now, what they are asking is calculate the number of generations the cells have grown in the given time span, considering there was no lag phase. So, they are telling that there was no preparatory phase for the cells. Consider that there are no preparatory phase. It directly goes to it starts dividing. As soon as you are inoculating it into a culture medium, this is an assumption. Okay. It starts dividing. And what I told earlier also during the session, that the life cycle of E. coli is 20 minutes. The life cycle of E. coli is 20 minutes. That means the generation time of E. coli is 20 minutes. That each one E. coli bacterial cell divides every after 20 minutes. Okay. Now, if we straight away take the question, take the thing that is being asked is how many generations of cells have grown here. Okay. Now, we don't need the initial number also in this case. We don't even need the time point and all. We just need 12 hours here. Okay. If it has grown for 12 hours and one cell divides every after 20 minutes, then what you basically have is 12 into 60, 12 hours. So how many minutes? 12 into 60 minutes. Okay. Divided by 20. So how this will give you the number of generations. So 20 minutes is one generation. So 12 into 60 minutes will have 12 into 60 divided by 20 generations. Okay. So 12 into 60 by 20 is what? 12. 60 by 20 is 3. 12 into 3 is 36. So answer will be 36. Okay. So basically this is here it is just asking you the number of generations. Generation time of E. coli is given here, which is 20 minutes, and the overall incubation time or growth time is given to 12 hours. So what you basically have to calculate is just the number of generations. You have to divide the generation time uh, from the uh, total incubation time. That's it. You don't need the initial number of cells here or what is the exact time point and all. Okay. So 36 generations will be the answer here. Now in the turbidimetric method, the OD of the bacterial culture is measured at. Okay. When we talked about this turbidimetric method, where is the method? So in this particular case, we saw that we are measuring the amount of light that is passing through the cell culture medium. Okay. Now, in that particular question, it's asking that in the turbidimetric method, what is the OD value? So the value that we usually get on this particular instrument, which is the spectrophotometer, it is called the optical density or OD value. Okay. So the optical density is measured at different at certain wavelengths where we are able to measure the growth rate of the cells or the amount of cells present in the culture medium. So for bacterial cells, we use it at 600 nanometers. Okay. Now also this is for bacterial growth we are talking about. Now sometimes after uh, we suppose have purified DNA, we have purified any kind of nucleic acid, we have purified proteins from cells. So that at that time also what is there is the absorbance or OD value. Basically how much of the light is absorbed by this particular medium versus how much was transmitted out of it. Okay. So this particular thing is called absorbance or optical density. So then for proteins, for measuring the absorbance of proteins, the wavelength used is 280 nanometers. For measuring the absorbance of nucleic acid in the cells or the nucleic acid content in the cells, the wavelength used is 260 nanometers. But for measuring the optical density or the content of bacterial cells in the culture medium, the wavelength used is 600 nanometers. Correct answer will be D. So the OD of the bacterial culture is measured at which particular wavelength? OD or optical density is measured at 600 nanometers. Okay. So this is it for today's class. Okay. We'll again meet in the next week where I will stop presenting. We'll discuss the next week's lecture. Just summarize and then we'll start discussing some example questions. Okay. See you all. Thank you for joining. Yeah.